Hi everyone, welcome to the channel. Dr. Hasi explains. This is Dr. Hasi, and today's topic is actually uh, into bio, and three chapters we are going to cover: chapter 12, 13, and 15, which are Mendelian genetics, chromosomal inheritance, and gene expression to proteins. So let's get started. Again, we are actually following the book Biology 2E, basically freely available online. If you just Google Biology 2E, you can actually see the link, and basically just you can download the whole book. It's an OER book, and it's actually the chapters you need to read, 12, 13, and 15. Chapter 12 is about Mendel and gene idea, or Mendelian genetics. As you know, Mendel is considered father of genetics because of his actually 2 papers and 8 years study on first experimental genetics. In 1865, published his papers and first show actually specific traits in plants like garden pea plants and how they actually move from generation to next generation to have 2 generations. And then he calculated actually ratios for specific number of genes, like monogenic 3:1 or dihybrid 9331. He did not study polygenic traits, but today we actually know there are polygenic traits also more than one genes. So Mendel used why Mendel's work so important, because he used this scientific approach to identify these 2 laws of inheritance, and then probability laws govern Mendelian inheritance. That means we can actually calculate these genetics of any situation by using probability and statistics. And inheritance patterns are often more complex. Sometimes they are more than just 1 or 2 genes. Mendelian genetics may not be able to predict, like polygenic situations. They are complex traits, more than 1 gene is involved, and there is a heavy environmental multifactorial situation going on. And many human traits are they can follow Mendelian genetics as well, and depending on the trait itself. So Mendel's work with garden peas provide evidence to replace the previous hypothesis, which was blending hypothesis, to the new hypothesis called particulate hypothesis, in which discrete heritable units he called, or factors, pass from parents to offspring. Today we call those factors. Today we call them genes. Okay, so Mendel's experiments, it was published 1865. Basically, there are examples here in this poster. You see a phenotype basically is the physical appearance of a trait or any organism, which is combination of genotype, which is the genetic makeup, and environment. And sometimes we see epigenetic changes like DNA methylation or RNA proteins can be modified by some other things beside these 2. So we will focus, this chapter mostly focus on genotype, which is the genetic makeup. It can be homozygous, like the same letter, big A, little a, or heterozygous or carrier, which is big A, little a. And gene can have several versions of it, which we call alleles. They can be dominant, recessive, a lot of time recessive, and sometimes we see incomplete dominant or co-dominant, like blood types, the AB blood type, which is co-dominant, we see, or incomplete dominance we see in some plants, like snapdragon flower color. But Mendel studied a lot of pea traits, like there were recessive, like green seed, wrinkle seed, white flower, short stem, and dominant for yellow seed, round seed, purple flower, tall stem, just some examples. And remember, plants don't have, they only have allosomes, that means male, female affected at the same time, same, and recessive always skip generation. So we call these type of situations autosomal recessive. From human situations like blue-green eye, attached earlobe, cystic fibrosis, sickle cell disease, albinism, Tay-Sachs disease. These are recessive and they skip generation. Dominant ones are like brown eye, widow's peak, freckles, Huntington disease, dwarfism, and myotonic dystrophy. These are some dominant autosomal disorders. They don't skip generation. And the polygenic, which means more than one gene involved, some examples from human: skin color, eye color, height, weight, diabetes, cancer, heart disease, some examples. And there is also mitochondrial inheritance, again, both male and female affected, and female transmit to everyone because it's maternal. Alzheimer's, ADHD, bipolar disorder, these are some examples. And then, of course, there is this whole, which we are going to see in next chapter. Also, let's talk about it now, which is X-link. So X-link examples are like, if it is recessive, they can skip generation still, but male more affected because male has only one X chromosome. Hemophilia, Duchenne muscular dystrophy, color blindness. These are recessive. We don't have many dominant ones, but there is one called Rett syndrome, which is X-linked dominant. Okay, so there are also situations that chromosomes, not the gene, but chromosome, can be actually, can have disorders itself, like aneuploidy, which means having 47 chromosome, and chromosome number 21 has an extra copy, which we know as Down syndrome, or polyploidy, like 3N or 4N type of situations. So let's look at chromosomes. Since we talked about chromosomal theory, which is chapter 13 a little bit. Again, we are following Biology 2E, the book, and basically this is chapter 13, chromosomal theory. Like last chapter, there was MENDEL in chromosomal theory. We also have a lot of new experiments, and Dr. Morgan, Thomas Morgan, another, a Nobel laureate, and that actually he came up with this chromosomal inheritance, and then studied actually fruit flies as his model system with his lab at Columbia University. So Dr. Morgan showed Mendelian inheritance has its physical basis and behavior of chromosomes. And he also discovered sex-linked genes that they might have a unique inheritance pattern. That means Mendel did not study this type of situations. So he actually worked on chromosomes. And as you see here in this picture, like a human, a male chromosome map, as you can see, 1 to 22 and XY. Each of them, this is called linkage map. This whole thing is called linkage. The reason it's called linkage map because each chromosome is a linkage, like linkage 1, 2, 3, 4. And genes, you see hundreds of thousands of genes in each chromosome. Largest one in chromosome 1, as you can see. Smallest one, Y chromosome, you can see here. Basically, if two genes happen to be on same chromosome, they are considered linked, and they might follow each other, and that doesn't apply to Mendelian genetics, so because they are linked. So always remember that. So chromosomes might be autosomes, that means 1 to 22 in this map, or sex chromosomes like XY. XX is female, XY is male in human. Chromosomes can be unlinked, that means they are on separate chromosomes. Genes on the chromosomes are unlinked because they are on separate chromosomes, or they might be linked because if they are on same chromosome, and they might have crossover and change parts, and we can calculate the combination percentage, and 1% of the combination is 1 MEP unit or 1 centimorgan. And that can actually help us to find location of the gene on that specific chromosome. Let's say we calculated it for chromosome 13, and we can actually find exactly, maybe it's in this band 20 centimorgan here in the shorter arm of this chromosome 13, possibly, one example. Then we write this 13p20. P is short arm, Q is long arm, and 13 is chromosome 13, 20 is this band here, exact locus of this chromosome. There are also chromosomal disorders because of alteration in number or structure of chromosomes. There might be aneuploidy, again, I mentioned, trisomy like Down syndrome, polyploidy. Polyploidy is pretty common in plants, and is but not in animals. And it can be a disorder in animals. So keep reading on these. Make sure that you actually look at the definitions, make flashcards of vocabulary, what's mono, poly, tri, di, and then look at some examples of each of situations from this chromosomal theory structure, right? So, and make sure that you know also some disorders like X-linked, like we see here, X-linked recessive human disorders like hemophilia, Duchenne muscular dystrophy, color blindness, or Rett syndrome as a X-linked dominant. We don't have really example of Y chromosome. The reason is Y chromosome is very, very small. There aren't too many genes on Y chromosomes. So this is so the chromosomes, and again, sex chromosomes versus autosomes and genetic disorders. There are some situations like dilation, duplication, inversion, and translocation also can cause genetic disorders. So this is chapter 12 and 13. And the last chapter is 15, which basically, this is gene expression. This was our last chapter on this module 4. Basically, this chapter, as you see in this poster here, basically, we talk about genes already. Gene is part of a DNA, and we talked about mostly nuclear genes most of the time, because most genes are located in nucleus. But they need to be transcribed to mRNA and then leave the nucleus, go find a ribosome and translate it to the amino acids and become peptides and polypeptides like proteins. That's our final phenotype, protein. So genes specify proteins via transcription and translation. Transcription is the DNA-directed synthesis of RNA. DNA-directed synthesis of RNA is transcription. And eukaryotic cells, on top of that, also modify their RNA after transcription, specifically mRNA or messenger RNA. Translation is the RNA-directed synthesis of polypeptide. And also mutations may happen, or one or more nucleotides can affect the protein structure and function. So a mutation in DNA can actually cause a totally different different nucleotide, and then they can actually change the whole protein. And and we've seen also how this energy, this information flows from DNA to mRNA to protein, right? So mutations can be nonsense mutations when a point mutation changes the amino acid codon into a stop codon and prematurely halting the translation of polypeptide chain and usually creating a non-functional protein. There is also insertion and deletions like we've seen in previous chapter. And mutagenes are the factors that they might be actually causing mutation, like we've seen here at the very first poster here, like here, some carcinogens or X-rays or viruses can be considered mutagenes. Okay, so keep reading on mutations in chapter 15. Make sure that you actually do your reading from chapter 15 from your book on both transcription and translation. Remember the enzymes. Remember actually how it's edited, mRNA edited, and then what's the final products, and then these specific details on mutations can actually change the final product. So we will next class we will actually continue our review. So this review ends here. Just to recap, this is actually module 4, which basically chapters 12, 13, 15, Mendelian genetics, chromosomal reductive Morgan studies, and genetic transcription translation proteins, which is chapter 15. So thanks for watching, and please feel free to comment, like, and share, subscribe to this channel, and share this video, and see you next time, and happy studying.